we're always really impressed by what the students are able to do in these Idea Accelerator programs in just a few weeks. And we're so glad that you've really embraced sort of these learn by doing, working with these synthetic biology ideas in practice and developing your own projects that really you originated, you did the research, you did some really fantastic work here.
Today is on malaria murders. We are the Holy Malaria team from Academy of Notre Dame.
Do you know how many people on Earth are affected by malaria? 229 million in 87 countries, 409,000 deaths. That's a lot, isn't it? Well, we have a solution. For our system design, our big idea was a system that causes the mosquito to self-destruct when it detects the malaria parasite. Our input would be histidine, which is a protein produced by the malaria parasite, and the output would be self-destruction of mosquitoes. Specifically, we chose the mechanism of alcohol production, which would kill the mosquito.
Our choice of chassis is mosquitoes. So one may ask, why mosquitoes? Well, there are many reasons. Firstly, mosquitoes are responsible for the devastating spread of malaria across the world. By altering the hosts so that they're unable to spread this parasite, we will be able to lessen the spread of malaria, therefore saving lives. Additionally, there is a personal aspect to this choice, as Ada, the group leader, aka me, is deathly afraid of mosquitoes.
For our parts and devices, we listed a couple that we think are necessary. Firstly, we need a histidine sensor that will sense the presence of histidine, indicating that the mosquito has malaria. This device should also be able to respond to the input by activating the production of alcohol. Secondly, we need a device that will produce alcohol. For example, some enzymes may be necessary for this production pathway. Additionally, we will need some DNA sequences to make this possible. We will need a sequence that encodes for the production of alcohol. One possible source that we thought of was yeast because of its abilities of alcohol fermentation. Pathologist from the Academy of Notre Dame. This is our presentation on an idea for a skin cancer diagnostic tool.
The topic area we decided to work on was medicine.
And so for system design, the system we designed should be able to serve as a diagnostic tool for skin cancer. This would be administered intradermally so that observations may be taken with the naked eye. The inputs would be irregular cell division in potentially cancerous cells, and the outputs would be color or pigment. And we chose the purple pigment, rhyolacin.
The next step to design this system was to choose the type of cell to use. We chose to utilize yeast as our chassis. It is a well-studied and safe organism, which makes it ideal for implementation in the skin.
Parts and devices. One part that we will need is a cell density dependent promoter, which will only trigger transcription of the pigment producing gene when there is excessive cell density due to the irregular cell division in cancerous cells. Another part that we will need is a pigment producing pathway to produce the output. We chose violacine, a purple pigment that can be produced by the bio-operon, which is available from iGEM. By linking the promoter to this gene, the bio-operon will only be transcribed when cancerous cells are present. Therefore, violacine, the purple pigment, will only be produced in the presence of cancerous cells.